away we go. So, hi, I'm Dave. I'm the county recorder for butterflies and moths for Glamorgan. I've been recording moths since the early 1980s and probably started seriously looking at micros in around about the early 1990s and around about around about 1995. So I've been doing micros for quite a long time as well. Come on, why is it not moving forward? Wrong button. Let's try that. Right. So I'm going to try not to refer back to last week's talk, which is available on the SEWBReC YouTube channel if you haven't seen it, but nevertheless there is a little bit of overlap. But yeah, I wanted to try and keep this as a relatively self-contained entity. But if we're going to talk about micromoths and how you identify them, we do at least need to define what a micromoth is. So yeah, it's not about size is the key message. So mother of pearl is a very large micromoth, and the marsh oblique barred is a very small macromoth. So it's not about size. It's all about taxonomy. And so this is the taxonomic order of Lepidoptera, split out with the red species being the micros, the blue species being the macros, the butterflies roughly in the middle. So just gives you an idea that in theory the macros are the more evolved and the micros are generally the less evolved, with the odd sort of honorary macro thrown in amongst the micros, and indeed the butterflies are thrown in amongst the micros these days. And when Phil Sterling and Mark Parsons produced their book on micros, Phil came up with this definition that the families that contain mostly large species are macros and the families that contain mostly small species are micros. And that kind of works, but that isn't necessarily very helpful if you've got a single individual in a moth trap, because if you're only looking at the one mother of pearl, you don't know that it's part of a family that have a number of small species in it. So that's not terribly helpful unless you learn the families. And the key message from this talk, as indeed it was in the previous talk on macros, getting to know the families is the key to being able to identify most moths, or at least get down to some kind of identification. Just to say that there are considerably more micros than there are macros, and yeah, and very few butterflies on the British list. So, and in the same way, again, as I say, I'm not going to refer back to last week's continually, but in last week's talk, I did say quite early on that I'm not going to be teaching you how to identify all of the 1,000 species of macros. I'm certainly not going to be teaching you all 1,600 microspecies. That is well beyond the scope of any single day talk. So yeah, this is more about giving you the information you need in order to try and get yourself to some kind of identification, or at least work out what group it is you're looking at. So this is the full list of families in the order of Lepidoptera, of butterflies and moths. And if we color code them according to the previous chart, with the red ones being the micros, the blue ones being the macros, and the yellow ones being the butterflies, you can see that if I was to go through every family, as I did with the macros, then it would actually take me quite a long time. So we're going to have to do things slightly differently. But I will take you through some of the families and give you some idea on where we can go on how we get to an identification. Now, the other thing, I do need to do a little bit of morphology. Let me just move. I've got you people are in the way, so let me move that. Right. Morphology. So last week I kind of skimmed over this, and we didn't really talk that much about the various parts of the body on an insect. But there is one aspect of the insect's head in particular that will be referred to quite a lot during the course of this talk, and that's the labial palps. So these are part of the mouth parts. There's two palps. The maxillary palps are tiny and not really very important, but the labial palps are quite a key character, and as I say, we'll be talking about those for several of the families when we go through them. The other parts of the insect that obviously we use for identification, the wings, the legs, the antennae, and the positioning of the antennae is really useful. And I won't need to go into hairy eyes with these. That's something purely for the macros. So again, so this is a stylized macro moth, but there are some key parts of the wing that I'm going to be referring to throughout, and I do use technical terms, and I would say that I would apologize for using technical terms, but you know, I learned these terms when I was 12 years old. I don't see why I shouldn't be passing that on to people these days. Purely an amateur, it has to be self-taught. So these aren't difficult words to get familiar with. So as far as the insect wing is concerned, it's vaguely triangular. The apex is the top right-hand corner in this instance, and the torus is the opposite angle. We don't really worry about the anal angle. We don't. Costa is the technical term for the leading edge, and dorsum is the technical term for the trailing edge, and termen is the third side of the triangle. And again, I'm not going to apologize for using these technical terms because there are times when referring to structures on the costa and structures along the dorsum it's actually a lot easier than using the cumbersome leading edge of the forewing and trailing edge of the forewing and whatever term they use for the termen. It's all a bit clumsy in my opinion, whereas costa is clearly defined as to what it is. The only other thing to really mention in amongst this crazy diagram is the cilia. So the cilia are the hairs that are the sort of hair-like scales that are found along the edge of the wing. And they're quite important for quite a lot of microspecies because they give the impression of extending the wing in a slightly different direction, and we'll come on to that in a bit. Venation is probably not a major concern, but yeah, it's worth bearing in mind. So when I started out, basically there were two books on that covered micros. There was Meyrick, which was published, this copy was published in 1926, and it was the only comprehensive book with every single species of British Lepidoptera in one volume. Fantastic book, really useful, and realistically the only way you were going to be able to identify the vast majority of micros, because there were no books on them. The other key book is the Field Guide to Small British Lepidoptera, which is one of the many publications that Maitland Emmet produced. And this was a follow-up to work that John Ford had produced back in the 1940s. Maitland updated it. And this book contains, again, all of the microspecies. It's just text, so there's no pictures, and the idea is that it gives you the life history information that you would need in order to find any given species. So it's split up into where you would find the eggs, what the larvae feed on, and what time of year you might find the larvae, when the pupa, or where the pupa might be positioned, and the time of year that would be around, and then some key characters on the adults, and where they are found. So it's very much a reference book, and it's a fantastic piece of literature, but no pictures. And I was quite fortunate as well to actually be in touch with a few of these eminent Microlepidopterists. So the picture on the left is Maitland, and yeah, sadly missed. Very influential and very knowledgeable Microlepidopterist. Came to it quite late in his career as well, so he only really started looking at them in his 60s. But by the time of his death, when he was, I think he was 92 when he died, he'd learned an awful lot and passed on an awful lot of information. And the chap on the left is chap on the right is very much still alive. That's David Agassiz, and he used to visit Somerset regularly because that's where his son was staying, and yeah, he used to pop down and say hi to our family and help us go through some of the micros that we were struggling with. So really useful contacts, and yeah, they were exceptionally helpful in my formative micro years. Now, these days, you guys are exceptionally fortunate. There is an awful lot more information readily available. The idea that you could go to a couple of field guides and have pictures of most species in Phil Sterling and Mark Parsons's book is fantastic, because there are quite a lot of readily identifiable micros. But then Chris Manley really sort of pulled out all the stops and produced his second edition photographic guide, which has very nearly all of the British species. It's very nearly a photographic equivalent to Meyrick, which is just a phenomenal piece of work. And I know that he's working on a third edition, and he's very much hoping to plug those final gaps and have what will be, the most comprehensive single volume book on British Lepidoptera since Meyrick. It's just phenomenal work. The other book that was produced a couple of years ago is an update to Maitland's Field Guide to the Smaller Moths of Great Britain and Ireland. So, because a lot has changed since Maitland last revised it, which was 1996, I think. So this was redone with John Langmaid, Steve Palmer, and Mark Young, and that was produced and published about 2 years ago, I think. And it's exactly the same format as the original, but all of this, all of the new species that have been added included, and yeah, so it's a fantastic book and essential, realistically, if you're going to be getting interested in micros out in the wider countryside, shall we say. So it's worth pointing out that moth traps are only really a small part of the picture as far as identifying micros is concerned. And indeed, when I started out on my path to micro enlightenment, I was employed in the Natural Museum of Wales, National Museum of Wales, and having access to the collections there was really useful. The vast number of species, being able to, so then when you go out into the field and you find them, it becomes a lot easier because you've already become familiar with many more species than you had access to in the books. And one of the contracts that I had with the museum involved going around South Wales with Darren Mann, collecting insects to bolster the museum's collections. And as a result of that, I came across quite a few species that hadn't been recorded in various parts of South Wales, and some indeed new to Wales, or at least so we thought at the time. And so I produced a paper. It was published in one of the entomological journals. Species, and I also included a line about the fact that we would have probably been able to record an awful lot more if we'd been able to do more light trapping. And Maitland Emmet decided that that was worthy of a response. So in the following edition of that journal, he added a, so he wrote that light trapping is a poor substitute to good field work. And that he issued a wager that he would spend one day in the field if someone was prepared to put him up. He would spend one day in the field and he would add 100 species to my list. So he came down to stay with me in Splot, and sure enough, we spent a day wandering around Plymouth Greatwood and planned our fields, and very easily got 100 species of leaf mining moths. And indeed, on the afternoon that he arrived, so before the actual day of the challenge, I think the first moth that we saw was Caloptilia rufipinella, and that was the first record for Wales. So it just really shows that yeah, there is no substitute for field work, but you have to know what you're looking at. And he was an exceptionally knowledgeable and charming man, and yeah, it was a really valuable day in the field learning micro moths from their leaf mines. But I'm not really going to be talking about leaf mines. I was charged with the idea behind this talk was to try and help people through how you would identify a moth based on the book by Parsons, Lewington, Parsons, Sterling and Lewington, wrong order, but you know the book I mean, and how you actually know where in the book to look. So this is the kind of thing that I was thinking of. You know, this is a small moth. It's brown and it's got a wingspan of about 9 millimeters. So how would you go through the book or the books and get a determination? Well, we'll come back to this moth a little bit later on. First, the flowchart. Now, as I said, I wasn't going to refer back to last week's. This will be familiar if you were here last week, but it's got a little bit of a tweak. So, when you first see a moth, is it one you recognize? And believe it or not, there are quite a lot of micro moths that you will learn to recognize over time. So it's not impossible to look at a micro moth and think, yeah, I know what that is. And if you do know what it is, or you think you know what it is, you go through the process of checking the books, and I'll talk about what checking the books relates to in a bit. And if the books confirm what you thought, then well done, you've got an identification, and hopefully your county recorder will agree with you. And the rest of the remainder of the talk. So if you do know what group it is, or if the books didn't confirm what you thought it was, then you look through the related species until you hopefully come up with something that looks pretty much, that fits what you're talking about. And if it does, go back, double check from the books, make sure that it fits the various criteria that I'm going to come on to in a sec. And if it doesn't, or if you don't even know where the group, what the group or the family is, then basically you're stuck with the task of going through the whole book, plate by plate. And at least you have a book that you can go through plate by plate, and it will come up with most things. So and this is where we diverge from last week's, what happens if you still can't find it? If you think you might have something that kind of fits, then go back and check the books based on what that came up with. But if it isn't, well, maybe it's not in the book, and it might be in the book, but it might be hidden. It might be in amongst the similar species. And if it's not, then the chances are you're going to have to work through some kind of key, or you're going to have to talk to an expert who can at least point you in the right direction. And it's worth bearing in mind that it may not even be a moth. Quite often you'll see these small creatures running around which look very much like micro moths. Well, in this case, it's actually a microcaddis, and the number of times that I've not only have I tried to identify these things that have been running around the bottom of the trap, you put them in a tube and you look at them with a hand lens and you think, oh no, it's a caddis. But I've also I get quite a lot of photos of these through, and yeah, there's only 6 or 7 species that you're likely to get of these microcaddis, but they're quite tricky to identify. So yeah, just do be aware of them. Handy hint to sort of identify that it is the caddis. They have hairs on the wings, not scales, and as a result of that, they do look a bit sort of scruffy. But yeah, once you've been caught out a few times, you'll get to recognize that these are not moths at all. So, checking the books. What is it that I mean when I say you need to check the books just to make sure that what you're looking at is what you're expecting it to be? And the first thing is, are there similar species that you need to take into account? Is the flight period correct? And the field guide of smaller moths will really help you with that. But as with other groups, flight periods can be a little bit unreliable, particularly as our climate changes and things come out at slightly odd times of year. It can still be a useful guide. Habitats and food plants, these are more important for micros than they are for macros. Micros don't tend to go quite so far from their food plants. So if you find micros in a moth trap, there's a reasonable chance that the food plant or the habitat is available not that far away. And similarly, is it found in the area? You do need to check your either the local moth group websites, or if they have an atlas out like we do, then check in there, or check with your county recorder. Some county recorders have a list of species that they would need confirmation of, and photos. And I mentioned, things nearby, and micros don't tend to move so far as macros, but of course there are migrants, and something like the diamondback moth here could turn up absolutely anywhere. It is a tiny moth, wingspan of not more than a couple of centimeters, about 1.5 centimeters maybe, and they fly across from North Africa without any problem whatsoever. So all of these things you need to take into account, that just because the books say it doesn't occur there doesn't mean it can't exist or can't be there, but you may need to get a photo or a specimen to your county recorder to double check that it is what you think it is. So how to identify a micromoth? As I said earlier on, the idea of this came about on where you go from if you have a moth, and based on that book, Sterling, Parsons and Lewington. And if you read the beginning of the book, there's actually some really useful chapters on, including a section on how to use this book. It's got a fantastic little key, which is nowhere near as complicated as the keys that I had to use when I first started out. And they've also got an at-a-glance guide, so you can compare the small moth that you have with the 104 species, little vignettes of, and see whether it gives you an idea as to what group it is that you're looking for, and then from there you can narrow it down. And of course, also within this book, there are species accounts for all of the species that it covers. It covers 1,033 species, I think it is. So it's a good, a good 2/3 of the microfauna, but it does have the caveat that there are families that aren't covered in this book. So if it's in there, fantastic, you can get somewhere close. If the family is completely covered in that book, then great. If not, then we need to do a little bit more digging, and this is where it comes to getting to know the families. So the micro families, here we go. There are quite a lot, and as I said, it's around about 50 families, and I'm not going to go through all of them, but hopefully we can tick off a few quick and easy families. So for example, let's look at these two, Elucidae and Pteraforidae. So these are the plumes. So the many plumed, there's only one, there's only one species of Elucidae, that's the many plumed moth. Some people call it the 20-plumed moth, which is a ludicrous name since it's got 24 plumes, so I always call it many-plumed moth. And it's distinctive and unique amongst the British fauna. The other plumes, there's about 24, 30 species of Pteraforidae, and in both cases, the wings are split up into distinct individual feathers. The pteraforids, generally speaking, some will roll their wings, like Lepidoptera and Emmelina monodactyla, but they all tend to sit in this sort of T shape, and as I say, Hexadactyla, the many-plumed, is pretty distinctive. Feeds on honeysuckle, very common, and these feed on a variety of herbaceous plants. So a really simple group to become familiar with, and importantly, all of the species are covered in Stirling, Parsons and Lewington. So plumes shouldn't really offer you too many difficulties, hopefully, although some of them can be a bit funky to identify, down to species at least. The next family to look at is probably the Tortricids, and this is one of the biggest families within the micros. So we'll take a little bit of time just looking at that. The palps are quite a useful way of recognizing that what you've got is a Tortricid. So this is a dissection from Chris Lewis's British Lepidoptera website, and these are the palps of this particular species, and they're forward pointing. The palps have 3 segments. So the first segment is a very short one, the second segment is this big hairy one, and the third one is another short one. And we'll see a few of these over the next couple of slides, but they just give the idea of a sort of a furry front to the face, and that's quite, as I say, the palps are quite important. There are 3 subfamilies within the Tortricids. The first is the Cochilids, and they feed mainly on Asteraceae. They feed mainly on what used to be Compositae, so things like sow thistles and things like that. And they all have, there's quite a few sort of yellow and creamy coloured ones. And they have a fairly sort of distinctive posture. They're all tend to sit sort of tent-like with their wings flat, sort of, roof-shaped. The terminal cilia along here, so there's the termen, and this section here is cilia, so that's not actually part of the wing. These are these hair-like scales, and they give it this sort of aerodynamic, almost sort of Concorde shape. And all of the cotylids will do that.
David, sorry to interrupt. If you're pointing with something...
Oh, I'm on the wrong screen. Good point. Sorry. Sorry, thank you, Roger. Yes, sorry. So that's the problem with presenter view. So this is the costal, the terminal cilia here on this species. So the wing actually ends around about here, and the terminal cilia are these sort of hair-like structures here, and it gives it this sort of Concorde shape, sort of aeroplaned.
Any folks?
No.
Sorry, just had to mute Roger there. Yeah, so that's the Cotylidae. The Tortricidae, then these are generally sort of broad, rounded along the costa, and they have a very distinctive sort of rounded sort of shape. They tend to be sort of relatively flat, but again with a sort of a gentle sort of rounded across the back. Very similar in shape. Some of them have these sort of the costa that sort of will come back out in a sort of sinusoidal kind of way, and some of them will have the males will have a what we call a costal fold. So you can just about make out on this one. There's a this here is a fold in the wing along the costa, and underneath there are scent scales. So only the males have them, and the costal fold can be quite a useful character for determining quite a few species of tortricids. The final subfamily in amongst the tortricids are the Oleothrutinae. These tend to be sort of halfway between the two in terms of roundedness. So they're not particularly flat, but they're not properly roof-like. They're sort of gently rounded as the wings roll around the body. Quite often have these what we call costal strigulae. So these are lines along the costa, and we'll talk about those in a couple of the other families as well. So generally rounded, sometimes flat across the back, and which across the dorsum, which gives the impression of a sort of almost squared shape, which again, once you become familiar with, you'll think, oh yeah, that's clearly a tortricid. And note again the palps. So it's just a big mass of scales at the front of the face. So yeah, so that's the tortricids. The next really big microfamily has been split into two, and I'm still a little bit rusty on where the split is between the Pyralids and the Crambids, but to me they were all Pyralids when I was growing up. So how do we tell that they're Pyralids? Pyralids are one of the bigger of the micros. They're well covered in the books, and one of the key things to note is where the antennae are. So they tend to sweep backwards across the back. So like this acrobasis here, they start sort of in the middle of the head, and they sort of go back along the middle of the wings and curve back out when they're properly at rest. So this female bee moth then it's probably not properly at rest in the same way as this thistle ermine. So, but when it's properly at rest, the antennae will be swept back across the head and across the wings. Now, as I say, the pyralids are a big group, so I can't really spend all day looking at all of them, but this hopefully gives you an idea of some of them. The other half are the crambids. Again, the antennae will be swept along the back. Again, he's not properly at rest, so he's they still give the idea that that's where the antennae are going. Big long palps. Actually, I should go back to the others. So some of these have big forward pointy palps, others don't. With the crambids, with what I call the original crambids, the grass moths, the palps really are very prominent and furry and forward pointing, very snouty. Similarly with the little grey ones, the Eudonias. So yeah, there's a lot in this group, and you really have no alternative other than just to sort of become familiar with them, I'm afraid. There's quite a few in here that will look similar to macros, so quite a few of the sort of the bigger Ananias or the Eudonias can be confused with some of the Aribidae, some of the snouts and things like that. I would also a passing mention to the Eudonias, the little grey triangular ones. These are very difficult to identify, and there's only 12 or so species, but half of them can't realistically be readily identified from external characters. There's a few. So Eudonia angustea is quite a distinctive one because it's kind of narrow and angular and pointy. So that's one of the few that you can readily identify, but there's quite a few in the group that you really can't. And if you're just starting out, I would be tempted to ignore those if I was you. So those are some easy ones to pick off. There's five big families of micros that aren't fully covered by Stirling, Parsons and Lewington. And indeed, they only really give them a sort of a passing nod. So I'm going to quickly go through some of those. I've lumped the Nepticulids and the Opisthegidae together because they do share some characters. Now, these are very, very, very small moths. These are the smallest of the British moths. And I would generally say that if you get one of these in your moth trap, you may not notice it. If you do notice it, you're probably better off just forgetting that you saw it, because it's generally far, far, far easier to recognize them as leaf miners, and leaf miners are definitely the key as far as these very, very small moths are concerned. It's about 100 of them, and they have these eye caps. So if I move on to they share this with the Opisthegidae, but it's a lot more clear on this photo. So this is the base of the antennae. It's covering the eye, and it's quite a distinctive feature of the Nepticulids and the Opisthegidae. Coleophoridae are one of my favorite microfamilies. Very distinctive sort of shape. There's about 100 species, so they have very long, thin forewings. The antennae are swept forward in a very characteristic manner, and the larvae feed in portable cases, which they make either out of bits of leaf or bits of seed, or in some cases just silk. Fabulous group, and again, the majority of Coleophoridae will require dissection, so if you're just starting out, you're probably better off forgetting that you saw them, or go and find the larvae, because the larvae are really funky, and I can't recommend that highly enough. The Gelechiidae are quite a tough group. There's 2 or 3 that are readily identified, but there's 40 odd species and most require dissection. Fairly characteristic resting posture with the wings sort of tentiform, and again the terminal cilia giving it this sort of tuft and making it look similar sort of shape to the cotylids that we saw earlier on. But if you look at the palps, they're not the big tufty palps that we had with the cotylids. They're more sort of swept upwards. Yeah, sort of quite long and forward sweeping. So Gelechiidae are quite a tough group, and again, I would probably not recommend you get too bogged down in them. So that's lenticulates. I'm going to jump around a bit. So the Gelechiidae, this is the group that was really problematic in my early days because there was really no literature on them at all. They are often, it's a big family. They tend to be fairly slim. They tend to sit with their wings flat across their backs. There are a few that don't. And they have fantastic palps, usually really long, pointy palps. And Anasia spartiella has the really long, pointy palps, but it also has these fantastic forward-pointing scales and hairs on the second segment. So yeah, really, really tough group. It's quite a lot that look like this, sort of small and brown and very nondescript. But again, there are some within the group that are readily identified. Anasia spartiella is one that you would potentially get to know. Now, the really key character to identifying Gelechiidae is actually in the hindwing, and it's the shape of the hindwing has this sort of finger pointing effect, which unfortunately you can't see on a live specimen. So really useful if you're taking specimens and setting them. So if you weren't sure what it was, and then you pull up the wings and you see this pointed finger, you think, right, it's definitely a Gelechiid. But from a photograph, you can't tell that, I'm afraid. The next big group is the Grassleariae. You can split it into sort of three groups. The Caloptilias are all in Stirling, Parsons and Lewington. The Pyralidae, one of them is in that group, and the Phyllorhynchus, there's about 100 and only 5 of them are illustrated. So the Caloptilias have this really sort of characteristic resting posture where they sit sort of like a dog sitting. So legs straight in front of them and giving this sort of triangular look. And there's a couple of related species. So the Aspeleptrix stringipinella, for example, sits in the same way. And they're all within this same group, and they're all illustrated in Stirling, Parsons and Lewington. So quite a useful group, most of which can be readily identified. Some of them require a bit of effort, but they're all covered in Stirling and Co. The Pyralidae also sit like that, but they're not as long, and these really are an absolute nightmare of a group to identify, which is why Stirling and Co only illustrated one of them. If you get one of these, then again, I would generally ignore it. You're better off going and finding the larvae, because the larvae are readily identifiable. Some of the key characters for identifying the Pyralidae are in the cilia, but on the underside of the forewings. So unless you set the specimen, you're really going to struggle to identify them. So yeah, probably worth steering clear of Pyralidae should you come across one. And the Phyllorhynchus, a lovely little group. Yeah, they're cracking little moths, sort of generally orange with these sort of white strigulae, dorsal strigulae along the dorsum, costal strigulae along the costa, and yeah, the little white streaks with little hooks, and they're just a really, really fun, useful group. And if we've got time afterwards, I'll take you through I've created an interactive key on the Glamorgan Moth Group website, which you can take some of these characters and help you determine which of the adult Phyllorictus you have in front of you, but I won't do that just yet. So yeah, this is another image from Chris Lewis's website, which gives an idea of the sort of shape. It's quite similar to the little micro that I teased at the beginning. So yeah, we'll come back to that in a bit. So that's quite a few families ticked off. 10 or so families done, but there's a few more to go. So let's have a look at some of the day-flying micros. So the most primitive of all the micros are the Micropteryxes. These are day-flying species. They actually eat pollen rather than supping on nectar. They're very small, with a wingspan of less than 1 centimetre. And in this family, the Micropteryxes are all covered by Sterling and Co. The Eriocranids, there's only 7 or so species. Peculiar sort of really hairy bodies, and there's only one that can be readily identified, and that's the golden one, which is the one that feeds on oak, and that's Subpurpurella. The other 6 are all purple, and they require dissection to be identified. So they do come to light, but mostly they'll be found swarming around their food plants. These all used to be in one family when I was growing up. The Adelids, the Incivarids, and the Prodoxids all used to be in the family Incivaridae. So these are your longhorns, of which you've got the metallic bronzy ones, and then you've got the dull brown ones. Most of the metallic bronzy ones are fairly straightforward to identify. The brown ones can be a little bit tricky. And then the others, the other groups, the Incivaridae and the Prodoxidae, they are small, sort of purplish brack with these dorsal marks and costal marks. The next group to sort of whiz through are, again, it all used to be in one family. It used to be in the Eponomutidae. They've been split out into 4 other families. Eponomuta evonimella here. So there's quite a few of these ermine moths, and indeed white moths with black spots is quite a common theme. And if you've got this one, which has a lot of dots, this is evonimella, the bird cherry ermine, then it's fine. But if it's one of the ones in the sort of the Padella Malinellus cagnagella group, then you've got to be really, really careful identifying them. Ideally, you need to know what they fed on as larvae. So it's the kind of thing that I would suggest you just ignore and let go, and don't waste too much time on. And also remember that there are other families that have white and spotty moths. So this is the pyralid, the thistle ermine. So yeah, it's superficially similar. Wrong way. But has a slightly different sort of resting posture. You'll note here that the antennae, for example, are clearly alongside the wing, not along the back of the wing. So Ipsolifer is, let's put both pictures up. I don't know why that was missing. So the Ipsolifer and the Ipsolifidae and Plutelidae, very similar sort of look to them. So this is a honeysuckle feeding Ipsolifer dentella, and this is the diamondback moth, the Plutella xylostella. Very, very similar, and indeed historically this species dentella used to be called xylostella back in the really old days. So even looking at Linnaeus's classification, there was some confusion as to which species he was referring to, and in fact it's been reassigned to this one from this one. Very similar, long and pointy, and they're all covered in Sterling and Co. So I was pointing at the wrong one, wasn't I? Long and pointy, forward-facing antennae. Quite a nice group to get to know. The Argyrestheas another group that are really easily recognized down to group level, because they have this peculiar posture of sitting on their front two legs, and then the final, the hind legs and the abdomen are raised and pointing upwards. So head down, bum in the air, you've got an Argyrestheid, almost certainly. There's a couple of others, but most of them are these Argyrestheids. And they're all covered in Sterling and Co. There's only about 24 species. So that's another group done. This next group that I'm going to look at are what used to be all in the same family. Again, it all used to be Echophoridae. And there's a few different groups. So the brown house moth and the white-shouldered house moth, big palps, sitting with their wings flat across their back, great. But then also in this subfamily, you've got something that sits tentiform. So resting posture isn't really very helpful with the Echophoridae, unfortunately. The palps are quite significantly important. And again, they're sort of recurved, pointing upwards, and quite tufty. The other two families, or two of the other families, sorry, include quite small families. So in this family, we have Dionaea flagella is a really readily recognizable species. It's one of the early spring species, and it's quite variable in color, but there aren't that many that look like it, and they have these sort of raised tufts. So this is definitely a micro that you can easily add to your just know them. And Carcina coarcana, once you've become familiar with it, it's not a species you're going to mistake. It's pink and yellow. There aren't many pink and yellow species. And it's kind of Tortrix shaped, but the antennae are much, much longer than you'd get in most Tortricids. And the palps are much more sort of big and pointy and not as tufty as you get in a Tortricid. And yeah, and it doesn't look like a Pyralidae either, again, because the antennae would start sort of on the back of the head and go trailing around the back rather than this sort of sweeping round. So again, Carcina coarcana is a species that it's common, it's widespread, and it's really, really distinctive. Once you've recognized it, you're not going to mistake it. On the other hand, the final family within this little group are the Depressarias, and there's two genera, the Agonopteryxes and the Depressarias, and they all have this, they're called flat bodies as an English name. They have this sort of broad rounded shape around the base of the costa, and in that respect they look a bit sort of Tortrix like, but they're flat. There's no hint of roundedness, and they have these fantastic massive pulps, which, as I say, Tortricids just don't have that. So that's how you would recognize them in terms of wrong way, in terms of identifying which one it is. The Depressarias are really tricky. They're all brown and grotty, and there's not a lot to them, whereas the Agonopteryxes, they tend to be a little bit easier. They tend to have distinctive markings that will enable you to get closer to the species, but yeah, they're not fully covered, unfortunately, in Sterling and Co, so you do need to be quite careful. Well, we've done quite a lot here. We've managed quite a few families. Let's pick off a few of the smaller families now. So these are all families that only have one or two species in. So yeah, we should be able to knock these out quite quickly. Lioneciidae, there's only two species, and one is exceptionally rare and the other is exceptionally common. So Lionecia clarkella, spelled that wrong, dear. There's no D in that. It's a very small moth, and you will see them as little silvery moths in the moth trap. But when you get a hand lens on them, they've got these fantastic sort of tails where they're sort of dark and sort of streaky. So yeah, the chances are you'll ignore them for quite a few years, but once you become familiar with them, you'll think, oh yeah, I can record those, and they're actually really easy. They're best recorded as larvae, as leaf mines. They have this sort of long meandering leaf mine, which is quite distinctive. But the really funky things are these, the cocoons. They create little hammocks on the underside of the leaves, and they pupate in those. Really, really funky. Love Lineacia clarkella. A little bit more tricky in terms of a group, the Pyralidae. Now, if you get the classic Praise fraxinella, which is this distinctive black and white moth, then they're fine. The problem is it also has a form that looks pretty much identical to this, which is Praise ruficeps, a very recently discovered species. And yeah, so these are quite tricky. These are really easy. And there's a couple of other species that have within this family that have just arrived in the UK that we haven't got in Wales yet, but found sort of in the southeast of England, particularly around London and Bedfordshire and that sort of area. Two more quick families. Bedeliidae, there's only the one species. Bedelia somnolentella it's a long and brown and thin, fairly distinctive species that the larvae feed on bindweed and are readily identified from the larvae. And the hawthorn moth, Scythropia cratagella, is another one that you will catch regularly in moth traps, particularly if you've got hawthorn nearby, and it's a quite distinctive grey moth. So white and grey, quite distinctive, but yeah. So that's two more quite small families. Right, we're getting there, because I'm not, as I say, I'm not going through all the families. Monthidae is another group, though, that is useful to become familiar with, because there's a few of them. They're common, and they sit with their wings slightly sort of rolled around them, and they have this sort of teardrop shape if you look at them from above. And some of them have these fantastic scale tufts. These are scent scales for attracting mates. So yeah, they're a fantastic group and quite tricky. There's only about a dozen species, but 2 or 3 of them you will get regularly in your gardens. Subistrigella is one. Monthodivicella is another one that you've almost certainly got in your gardens, but there are a couple of similar species that make them quite tricky to tell which is which. Getting there. Tineidae is the next group that I'm going to quickly look at. These are detritus feeders, primarily rotting fungi and rotten wood. They tend to sit very much like the cotylids. But the head is really quite sort of furry. The palps are a little bit more sort of upright and a little bit more furry, but otherwise they are superficially quite similar. There's quite a few tineids. These are includes your clothes moths and things like that. And yeah, can be quite tricky, but there's some that are really easy. So Nemopogon clementella is a really distinctive moth. Once you've realized it's not a Tortrix, it is this Nemopogon, then which feeds on wild clematis. Yeah, you're getting there. So the final family that I'm going to look at is Bucolatricidae. Again, they have these big sort of eye caps. So similar in some ways to the Nepticulus, but they're much bigger, much longer. They have these really funky hairdos. They sit more sort of tent-like, and yeah. So and you've noticed that the costal cilia isn't always in the same direction. So this one, which is Nigrica, which feeds on oxeye daisies, the costal cilia is swept along the wing, so it makes it look even longer. Whereas on Bucolatrix siderella, it's a little bit the costal cilia sort of sit up, making it sort of shorter and yeah, more sort of plane-like. So let's come back to that mystery moth that I showed you at the beginning. What can we say? Well, we can knock out a few of the species. It's clearly not an epticulid. It's long and thin, and the head's wrong. It's not a Coleophora, for example. The antennae are swept back. It's clearly not one of the big ones like the Tortricids or the Pyralids. Could it be an Elechistid? No. Elechistids have more prominent palps, and the hairs on the head, that they don't have this tufty head. It could be a Phylloricta. It's a bit big for most Phylloricta species, but the head would fit. But no. The thing is, I already knew what this was when it appeared, because I'd actually reared it from its larval food plant, which was C. aster, and that makes it Bucculatrix maritima. And how would you have identified it? Good question. If you hadn't reared it from the C. aster, you'd have really struggled, because it's not in Stirling, Parsons and Lewington. So yeah, you just have to get to know the groups to recognize, well, it's got a big tufty forehead, it's a long thin thing, swept back antennae, maybe it's one of these other groups, and then you'd have to just work through as many different groups as you can. Not that easy. So yeah, Bucculatrix maritima. And why have I got multiple pictures of that? Right. So, just to summarize, micros are quite tricky. They're diverse, they're complicated. There are quite a few out there that fit into the just know category once you've become familiar with the various groups, but there's an awful lot that aren't. An awful lot where just you've got no idea where to start, and that's really where you have to become familiar with the families, work out what the characters are that will take you to the family as quick as you can. But really with micros, the larval stages are the key, and if you really interested in getting into micros, find the larvae, look for the leaf mines, and that way you know what it fed on, and that helps considerably in determining what species you've got. Don't rely on moth traps when getting to know the micros. So thank you to everyone whose photo I've used. I think I managed to credit pretty much everyone this time around, unlike in the previous talk. So yeah, so that's that as far as today is concerned. So I'm going to stop the share for now. And as I say, I'm prepared to demonstrate my Phylloricta interactive key if people want to see it, but I'm not going to force everybody into seeing that, so we'll leave that for the hardcore at the end if people really want. So were there any questions, Elaine?
I am checking now. Molly Kirk asks, is this the same terminology for butterflies as well? I think that was talking about the names for the different parts of the wing, etc.
Yes, they're not so much used in butterflies in terms of what most people. Yeah, but they are the same characters. They are the same names. It's unique, in fact, amongst all insects. It's not just butterflies and moths. So yeah, we use them across all of them.
Jan Miller asks if all micros are leaf miners.
No.
That would be too helpful.
That would be far too easy. Yeah, no. Certain families will be leaf miners. So the things like the Nepticulids and the Grassleariae, the Phyllorictas, they're all leaf miners. The Coleophora are sort of partial leaf miners in that technically a leaf miner would spend its entire life within the leaf, whereas the Coleophoridae, they move around from leaf to leaf, but they leave these blotches on the leaves, and indeed some of them eat seeds. So they're not leaf miners, they'd be seed miners, I guess. But then there are others that will graze. So things like the Buculatrix group, they start off as leaf miners. So in the first couple of instars, they'll make little mines, and then they go on and graze. So things like Buculatrix thoracella, or the one that feeds on lime, you'll quite often find the sort of the grazing marks of the later stages. And if you keep looking, eventually you'll find a little sort of angular leaf mine. So yeah, so no, micros aren't all leaf miners, I'm afraid. It's not as easy as that.
Andrew Cormack asked if there is a good reference book or on a website for leaf miners.
Yes, for leaf miners, go to UK Leaf Miners, I think it's called. Hang on a second. British Leaf Mining. I'm going to share my screen again for a sec. So, where are we? That one. Share. So the British Leaf Mining, it's called British Leaf Mines, leafmines.co.uk. It's a fantastic resource for leaf mines, and it doesn't just do leps. It covers all insect leaf miners, because flies, beetles, all sorts of other things do create leaf mines. And if you know your plants, so if you know the plant species that you're looking at, you can go to the plant key and it will take you. So for example, impatiens, there's only one species that feeds on impatiens. It's a fly mine, and it's horticola. So there you go. So that's a really, really good website for British leps. There are a few others that are worth looking at, but this is probably the most comprehensive and the simplest answer to give at this stage.
Cool. Andrew Bevan asks if there's any recommendation on hand lens magnification level.
Depends on how small the moth is, I suppose. I don't tend to use anything more than a ×10 for most of my in the field work. But there will be times when the moth is considerably smaller than that. So you probably want to be looking at maybe ×20, but I wouldn't go too much more than that. ×10 is generally quite adequate in the field.
Paul Sutherland's mentioned that he's used iNaturalist to get some identifications. So you could use that as a starting point if you weren't sure where to go in the book.
Yep, fair enough. It's not a system I'm familiar with, but yeah, I've heard it mentioned a few times.
Is there a book that covers? This is from Neps. Is there a book that covers food plants and species of Lepidoptera that could be found on them? I never know where to start with the larvae.
Yeah, well, for the micros, then you can't do any worse. I didn't bring it across with me, but the smaller. Let me go back to this one. Where are we? Where's it gone? Books, that slide. If I go to there, nope, that's not gone back to there. The John Langmaid, Steve Palmer and Mark Young book, that one. Back to Zoom, back to share screen, and that one, share that. Yeah, this book here, the Field Guide to Smaller Moths of Great Britain and Ireland, is the comprehensive guide to your micro moth food plants. It's got an index, so it's got a food plant index as well as a moth species index. So if you know what your plant is, it will give you all of the key codes for all of the species that feed on that food plant, and from there you can just rattle through them until you know which one it is. And if you read the information within the guide, it will also then provide you with the field notes, so you'd know, for example, if it's the Coleophora, it's feeding on the seed head, not in the leaf mine. So if it's on the leaf and it's a mine, then it's probably one of these species. So it is by far the best book to get for micros. Doesn't cover macros. If you want to do macros, I don't think there's the same kind of information readily to hand. There are some really good websites. Not that you can think of one off the hand. Reg Fry's website was really good. UK Leps, but I don't have the link to hand.
We'll send an email after the course and include all the links to relevant information.
Yeah, there is also
by then.
Yeah, there is also a website for all of the food plants. I'm sure there's a larval food plants website again, which covers not just leps, but it covers other insect orders as well. I'll have to dig that up. I can't remember where that is.
Okay. Emma Williams asks if there's any tips for photography. One of her major issues is using a flash and the colours appearing really different.
No, most of the photography I do these days is using my phone. We kind of need someone like Graham Watkins on the line, I think. Is Graham here today? I don't think he is he? No, I'm not an expert. Well, Chris Manley would be the other person to talk to, I guess, because his micro photography is fantastic. He uses a ring flash for most of his stuff from what I remember. But no, I'm afraid I can't really help you there, Emma. Sorry.
I'd say to pot stuff and pop them in the fridge to calm them down can be a useful starting point because they're still then.
Can help, yeah, it certainly can.
Phil Smith asks, I occasionally come across larval stages of moths in aquatic habitats. Is there anyone in the UK who can identify them? I seem to recall they're all in the family Crambidae.
Yeah, they're the China marks. David Agassiz is actually a national expert on China marks. He did some work on the China marks that you found in certain glass houses, and added half a dozen species to the British list because they were found exclusively in these glass houses. In terms of how you get to identify them, it's not a group I know particularly well in terms of identifying them, but I would imagine that the book that I've mentioned several times will tell you what if you know what the food plant is, then you've got a good chance of working out which species it is amongst those China marks. Otherwise, try and rear them out, I guess. Although, again, that's not going to be easy unless you've got a fish pond, I guess. Don't know. I don't think I've answered that very well at all. Sorry.
So there is a particular person that.
Well, David Agassiz would have. Yeah, I mean, but I don't know whether it depends on what information you've got in terms of if you've got a photo of the larvae, then I'm not sure whether David would be able to answer. In fact, it might be worth checking on the UK Moths website, because I know Ian Smith took a lot of larval photos and put them on there. So don't know. As I say, it's not a group I know that much about, other than the versions that come to the moth traps, I'm afraid.
Jan Miller says when she first started to learn macro, she was told that thin body is a geometrid, fat body is a noctuid, and that helps you cut down when you're working out what family it is. Is there a similar guide for micros?
Not really, because there's too many families. No, when I started out, it was relatively straightforward. We only had books on pyralids and tortricids, and that was pretty much it. So you went through those books and you became familiar with the pyrals and what is now crambids, and you became familiar with the tortricids. And if it wasn't in those books, oh, someone's got. Thank you. Someone's showing there for their cameras. Yeah, so if it wasn't in those, I got to know those groups, those families really, really well. And from then, if it wasn't in one of those families because it was longer and thinner or sat in a funky way, then you knew that it was you were looking in one of the other groups, and then you were going through the various sort of pamphlets, because that was all we really had. So other than the families that I sort of touched on, things like the Argyrestheas that sit with their sort of head down, thumb in the air, or the Caloptilias, which are head down, the other way around, bum down, head in the air like a stood dog, then you just have to sort of get to know them. Funky hairdos, position of the antennae, these are all sort of clues, but it's too diverse to say that it fits all species, if that makes sense.
Okay. Ian Spence says, is anyone working on external ID, even with a microscope rather than dissection?
I think for so many groups, there are external characters, but they're external characters that that aren't necessarily reliable enough to be able to say which one the species is. So dissection will always be the fallback for quite a lot of species, particularly if they're slightly worn, and that's just the nature of this level of entomology, I'm afraid. So yeah, I mean, you know, there's no doubt that when I started out that you needed to take specimens for an awful lot more species because there weren't the images, there weren't the literature available. You had to be able to take your specimens to someone to be able to show them what they were. We didn't have digital photography to that extent, and you know, and the photos wouldn't necessarily be of good enough quality to really help. So you kind of had to get into the habit of taking the specimen if you really wanted to get to know what it is. It's not the same now. There is an awful lot more information out there, and there are an awful lot of species that can be identified from photos, but not all of them. So yeah, it's got better in that respect. And certainly you can do an awful lot of micros without dissection and without taking specimens without any problem whatsoever. Yeah.
Cool. A couple of people talking about photography. So Mark Sheridan said, does he uses an Olympus TG5, which has great onboard stacking facility. Mandy Ford recommends using a diffuser in front of the flash or using a ring flash, and also recommends Northumberland moths for larval food plants.
Fair enough. Yep. Is that Paul Talbot? Ian Tams. Yeah. Okay.
Emma's put a really long message, which I won't read out. More ring flash. I think that's it for questions. If anybody wants to ask a question verbally, you can unmute yourself and ask a more complicated question if you can't go up to type. But if we don't have any other questions, do you want to showcase your screen, Dave? So if you're not hardcore, you can leave at this point.
So right, let me share my screen again. Let me bear with me a moment. Get rid of that. Get rid of that. Share. Which one is it? It's that one. So this is a visual key. It's based on technology that Rich Berkmar created. And it goes through, the idea is the more of these items that you can tick off, the better chance you've got of getting an identification. So, for example, the ground colour, the majority of Phylloneuropters are orange and brown. There's a few that are white. So if I select that one, then it's the list of species filters. So the ones that are white are the ones on the right, and the ones that are orange and brown are the ones on the left. So the higher the score, the closer match it has to what you're looking at. Costal strigulae. If you count the number of little white marks along the costa, and you've got, say, 4 costal strigulae, and then along the dorsa you've got 3 dorsal strigulae, then the ones that match that have now got a score of 3. The ones that don't at all have got a lesser score. So anything that's got a score of 3 is still in your list of things that you could include as your species. The fascia, there are little helps. So does the wing have a complete line? That's a fascia there. and this is a species that does not have a complete fascia. So if your species has a complete median fascia, at least one, then again it reshuffles. So now we're down to 4 species, sorry, 6 species that are on a score of 4. And if you happened to know, or you beat the thing from a particular species of tree, then you could say, oh, I think that was on willow. And that leaves you down to 2 species. So it can really help sort of narrow down the kinds of things that you're looking for, and give you a starting point to then go and look at the particular species. So food plant fascia, apical hooks. These are in the I've only got one image. That's a shame. So the apical hooks are lines within the costal, within the terminal cilia. So this is supposed to be showing. Yeah, there it is. This is type 3. I had thought I had images for all of the types of hooks, so excuse me, I'd better correct that. But yeah, this is a type 3 hook. So if you could see that within your moth, then you could tick that in there. And the medial line, this is a line across the middle of the thorax. So in this case, this has clearly got a white line across the middle of the thorax. So that would, if you would tick that as saying yes on there, and that would filter again. You can include things like wingspan and the presence of a basal streak. Actually, if I go back to that, this is a basal streak, this line here. Let me have a look at some of these images. A lot of the images that are in here are from Chris Manley. Yeah, so there you go. That's an orange moth. That's a white moth. It's as simple as that. So yeah, that's kind of that's the sort of the 2-column version. There are other versions, so you can do it from different formats, so you can see which species scores on a particular thing, and from this view you can then see, oh, there's a photo of that. So was it Trifasciata? No, it wasn't that species. Was it Regella? Oh, that's a nice basal streak there. So that's, as I say, the Phyllorictors they're probably more easily identified as larvae, but if you've got an adult, you've got a hope of at least getting somewhere close using something like this key. You can also get the full details of species Lanternella. Let's go in, or Muellerella. There you go. So there you go. Gives you all of the information that's within this test data set, and if there's a photo, it will give you the photo. Okay. There you go, little whistle-stop tour of my Phylloricta Tom Bio visualization key. There's another one for pugs, which is reasonably useful.
Just to say, it's a Field Studies Council product. It was, yes, yeah. That's right. So it was developed as part of a Field Studies Council project, and you can make your own. If you know a group really well and you want to make your own key, you can use that tool. Yep. So if there's no other questions, I think we're done a bit earlier than last week.
Yes.
So thank you very much for coming, everybody.
Indeed, thank you very much.
And yeah, I'll email everybody probably tomorrow or the day after with the link to the YouTube video and any links to books and stuff we've talked about today. But yeah, thank you for coming.
Yep, thanks everyone. Hope you all found it useful.
Yeah, thanks. Cheerio.
Cheers.
Thank you.